Okay, I think we'll get started. The rate of clicking in of people has slowed. Hello, everybody. So lovely to have you join us for our second Global Biofoundry Alliance webinar. We intend to hold these about monthly, and the idea is that we are rotating globally, so through the time zones, that we can truly get buy-in from all of our members and also from all the interesting work that's being carried out across the world and within our network. Today's seminar will be recorded, and for anybody who'd like to revisit or to view it within a better time zone for yourself, it will be available on the Global Biofoundry Alliance website. It's lovely to have you join us. Thanks again for your time. Today we have two great speakers from our region. Chueh Loo Poh from the University of Singapore, the National University of Singapore, and Chueh Loo is going to talk about developing open source software for synthetic biology. And Esteban Marcellin from the University of Queensland will be speaking second, and he will be talking about the collaboration that the University of Queensland has with Amyris. So why don't we get started? Sorry, Chueh Loo, housekeeping, I would just say the Q&A window should be open. Does someone want to shoot a question and we'll just test it out? The Q&A window. Hey, great, thanks, Nathan. Perfect, so the Q&A window is open. If we use that for our questions, perhaps the speakers can answer them at a later date if we run out of time, or Chueh Loo can do it during Esteban's talk. So yeah, you can also raise your hand, but why don't we stick with the Q&A for the time being. Okay, well, Chueh Loo, please take it away.
Okay, thanks. Let me start sharing my screen. Hope it works. Okay. So I trust everybody can see my screen.
Yes, no problem, Chueh Loo.
Okay, good, good. So thanks Natalie and Matthew for inviting me for the talk. Right. So yeah, I'm very happy to be here to give this seminar. And so I'm from NUS, as what Natalie mentioned. At the same time, I'm also the co-chair of the GBA software working group, working together with Nathan and Evening. So today, what I plan to talk about is really some recent effort that we have been doing at the GBA software working group, where we try to develop a standardized open source Python software package that we think will be very useful for the biofoundry operation. And at the same time, I'll share with you guys some of the recent work that we are doing at SynCTI as well, where we develop tools to help us with enzymes engineering, and also how we can go about developing software models to help us in the genetic circuit design. Okay, so I hope you find the software tools that we are going to talk about useful for you. Okay, and I'd love to hear any feedback that you have for this solution. So without further ado, let me start with the context of why we are doing what we are doing. So essentially, I guess you guys are aware, we are facing a lot of problems now, relating to climate change. We have this growing population, particularly with the aging population. And then with that comes with a huge demands for goods, for food, and then therapeutics, right? Then at the same time, so we're also generating lots of waste, right? And then we also see a drop in the amount of fossil fuel sort of resources available for us. Essentially, the current linear-based economy is really not sustainable. And then on this note, right? So that's where we feel that the engineering of biology is actually playing an important role. And to me, the reason is that through engineering biology, it gives us the potential to utilize renewable resources and to produce a range of materials that we need, right? And it also gives us the opportunity to create what we call the circular bioeconomy kind of approach, right? And with that, we see that it has great potential to address the issue that we are facing right now, based on the linear economy. However, as you know, when we engineer biological system, right, it's very complex. It takes a long time. It takes a lot of resources. And that's why in the recent year, we start to see the emergence of biofoundries around the world. It's not just part of the region, but it's across the world. So essentially, biofoundries are labs that are equipped with advanced automations and high throughput equipment, supported by computer-aided design software, to help us to really carry out rapid prototyping of a biological system, right? So essentially, the biofoundry can be considered a key to this new bio-based industry. So essentially, to me, the biofoundry really provides the integrated infrastructure to bring us across the design, build, test, learn cycle, which is now quite a common framework that we have been using to engineer our biological system. Biofoundries are equipped with automation to help us to go through this cycle rapidly. At the same time, it provides high throughput assembly of DNA, help us to test many cells very rapidly. And some of the biofoundries are having integrated analytics and omics measurements platform as well. And last but not least, software is also part of the integral part of what the biofoundries offer. Right. And because biofoundry now can actually generate huge amount of data, we are starting to see more application of using sort of the AI and machine learning in going through a more data-driven learning approach, which is to essentially help us to design our biological system better, right? So this is kind of like a quick snapshot of what a biofoundry entails. Right. So what I'm showing here is really a bio-automation platform that we have at SynCTI at NUS in Singapore. So essentially, we have a whole pipeline of liquid handlers to facilitate how to move our samples around. We also have an automated colony picker. And one of my favorite is really the high throughput measurement mass spec system that we have, that can bring us through a very fast mass specs measurements. It's a system by Agilent, right? It's the rapid fire. So essentially, this is just one example that we have, in Singapore. I believe, around the different biofoundries, they have a different setup, right? But essentially, that's this whole sort of automation pipeline, right, that bring us across the high sample preparation. So in May 2019, right? So we formed the GBA, in Japan, right? So I was just looking at a photo. It was such a nice things to have when we still be able to come together and take that very nice photo. I missed that time. I hope one day, very soon, we'll still be able to come together and take that photo. But anyway, so during the launch of the GBA, with several foundries decided to come together and to work on common challenges, right? And one of the common challenges that was identified, in fact, was software. So we see that software solution is actually vital, to support the various specialized tasks along the design, build, test, learn cycle. And this is really because, if you imagine the number of samples that's in process in biofoundries, these are like, in the order of like 10 to the power of 2, 10 to the power of 4 kind of magnitude. We really need the software to facilitate the handling of all these samples, right? So the whole idea is really, can we try to reduce the errors that is generated if we manually have to process all this data? We believe that software is key to help us to reduce all these manually generated mistakes. Because of this large number that we are handling in the biofoundry. So this kind of mistakes is can be really costly, both in terms of time and money, right? So as such, we now see actually a lot of software tools available to many of the researchers now that covers the wide spectrum across the design, build, test, learn cycle. So I'm just going to give a few snapshots on the software tools that's available. But I have to say they are not exhaustive. They're just some examples that I am aware of, right? So during the design, we have tools like Cameo and RetroPath that actually facilitate the metabolic engineering design. We also have the EFI in Singapore to facilitate us in discovering novel enzymes and going around the design, build, test, learn cycle, right? We also have DNA chisels and part genie to help us to do sequence optimization. And coming to the build stage, we have this array of software solution as well, like Conflow, TestElegend, and Synthesis. These are all very useful and powerful tools to have, to help us in managing how we go about doing the build part of the biological systems. Then we also have softwares that manage the data analysis, right? So this MZMine is a tool to analyze the mass spec data. And last but not least, coming to the learn phase, we have modeling tools like Cobra and the tools that we are building in SynCTI, to help us to create models to understand our system better, to gain more insights into how the system behaves in addition to all the experimental data that we are getting, right? So what we are seeing here is a whole range of softwares that are available right now, to all of you. So before I go further, I'd like to share what we have been doing in SynCTI, 2 platforms, and we believe that you might find them interesting and useful. So first of all, we have this EFI, being hosted in Singapore. So this EFI is actually an initiative founded at UIUC. So essentially, it's a database and software tools based on the similarity network to discover novel enzymes. Right. So we actually coupled this within the Singapore Biofoundry to help us to discover novel enzymes based on certain characteristics, and then going through the foundry to do testings, expression, and then essentially trying to customize the enzymes for food processing. So this is a platform that is really driven by Wunshan at NUS. So this is the first platform I want to talk about. For the second one we are building is really this software tool. We call it BMSS. Essentially, it's a software that helps us to identify models automatically, for genetic circuit design. So essentially, what we like to do is to have a model to gain insight to how a particular circuit is working. But then we always face with this problem. So how should we go about, say, for example, these genetic circuits? How should we go about building the model for this? So conventionally, what we're going to do is to write equations, right, starting from the first principle. And we have been doing this in a very repetitive manner. So then one day we thought, what if there's a system that, if we can pump in the experimental result and voila, just come up with the models that we need, right? Wouldn't that be fantastic, right? So based on that motivation, then we decided to build a system, precisely to do that. So essentially, we built this BMSS to take in experimental data, for example, genetic circuits like an AND gate. And with that experimental data and maybe some characteristics about the gene circuit, the system will actually go through a database, we call it a model base, to find the best fit model that can best describe the experimental data that we are talking about. And eventually, then we can use the model that's output and then use some other tools to help us to further analyze the model, right? So we already have this as the first version, but then we actually move on to try to improve this. So more recently, we have built up the BMSS 2.0, right? So essentially, we make the first version more flexible, we create more modules, and we make it easier for the user to input the models to build up this model database. Essentially, to us, the key is actually having a lot of models in this database so that the software can go in and try to search, right? At the same time, we also incorporated a few more analysis tools that is less commonly used or available in the conventional modeling software tools. So this tools is actually available in GitHub as well. And we welcome you guys to use. So essentially, we are using BMSS to really test hypotheses rapidly once we have the experimental result. Then at the same time, we are incorporating this software into the design, build, test, learn cycle. Essentially taking data rapidly from the test phase that we can quickly create the models and to analyze them and then to help in the next phase of the design, right? That's what we wanted to do, right? So literally to know, in addition to looking at experimental results, can we learn more from the models? And then coupling the two knowledge together, we hope to speed up this whole process of the engineering of the system. So essentially, I've covered like two systems that we have at NUS, right? But essentially, there's really a wide range of software tools that covers the design, build, test, and cycle. So thinking about this, we should be pretty good with software, right? Since there's so many software out there. But actually, we still have a problem. And what's that problem? The problem is really to select the appropriate software. It can be difficult. It's like there's so many choices, right? And then all these solutions are really very scattered. And then we don't really have a very definite guidelines to how we should go about selecting the software. Usually it's through word of mouth. We start asking people what is good and what you're using. And what we realize is that if you talk to 10 people would you be using like several different software. And there's no universally agreed state of the art as well. There's no one clear state of the art that we should use. And what's the consequence of this? So we start to notice people start to develop similar softwares, right, for similar challenge, to solve similar problem. And we see a lot of effort being done in trying to recreate the wheels, right? So there's a lot of wheel reinvention. At the same time, because these solutions are developed within a lab, they tend to be very specific to the requirements of the lab. And that makes it very difficult to reuse those solutions, once we want to apply that to a different lab. At the same time, we also notice several solutions out there. They tend to have an end-to-end application in mind. And that also makes things difficult to sort of unplug certain modules within that end-to-end application, right? For use in their own software. So this is the kind of situation that we face when the software working group comes together. And then we look upon another community that actually we realize that they face a similar situation. And that's the bioinformatics community. And in fact, they faced similar situation back then. And what they did was that they introduced this package called the BioPython. In fact, it's a software package based on Python with a number of libraries, that can help the community to actually analyze their data. Right. So it's actually a community-driven activity here, right? And what we learn is that having such a package that's open source, it actually helps to increase reliability of the software because the community is testing it. And then at the same time, it increases the reusability and also the interoperability of the software as well. Now, at the same time, we also realize that there's more community engagement, right? And the uptake is actually higher because all the software is actually housed under one common umbrella, right? So it's easier to discover whether there's any new features being introduced. And then we see that this is a very successful sort of a package out there. We see that this package has been cited by more than 2,000 manuscripts, and it has over 3,000 GitHub projects, using BioPython. So we are very inspired by this. As such, in the year 2019, when the software working group was deciding what to do, we decided to learn from this BioPython and to introduce what we call Synbio-Python. So the reason is that we realized that the BioPython is really catered to the requirement more for the bioinformatics community. It lacks that kind of more sort of a package for synthetic biology sort of activity, right? Like DNA assembly and automation, right? So we thought maybe we can have a package, in a similar kind of mode, but more catering to the synthetic biology requirement. And with that, the software working group came together, and there are several contributors to this whole effort. They are all shown here. So they came from all different biofoundries, as you can see from this part of the slides. We have people from SynCTI, from Manchester, from Edinburgh, from Finland, Liverpool and Australia as well. So it crossed several time zones, right? If you can imagine. So this whole piece of work that we have been doing for the past year is all done online. The only time we can come it's really through Zoom, multiple Zoom and Skype sessions. And it's amazing what can be achieved, even during the lockdown and the whole COVID stuff, right? So we spent the whole of last year trying to develop the software. So now it's already available on GitHub. At the same time, we have put forward the manuscript, which has also been recently accepted. So I thank all of them to contribute to this Synbio-Python actually. So let me now briefly talk about the various modules that's currently available in Synbio-Python. So essentially there are 3 of them. So the first is Gene Babel, which allows us to sort of translate the different file formats that's in synthetic biology. And the second one is the automation library. And the third is the codon usage. So I'll just briefly go through what each of them does. So essentially, Gene Babel is a repository of a standard file puzzle, right? As you know, there's already a number of standard file formats already available, and they are there to capture information at all levels, right? So we have GenBank files to capture gene circuits, gene sequence informations. We have SBOL that capture genetic circuits design. We have SBML that captures the models, right? So we can see that there are several tools that's already adopting all these standards. But what we see is lagging is that there's no one size fits all kind of tools out there that can support all this conversion of all these different file formats. So then we decided to create this particular repository where we start storing all these different file puzzle that anyone who is interested to have a puzzle can come to this repository and have that tool available. So then the second sort of modules that we have is really the automation library. We hope to provide an easy to use standardized solution to create automated workflow, right? So we acknowledge that there's already quite a number of software solutions out there that can actually create all this automated workflow. For example, Aquarium, Enter by Syntace, Tesselligence, and Auto Protocol. So these are pretty well-built, very sort of a powerful package actually. But what we also realize is that some of this software doesn't allow the developments of protocols via scripting, which is something that developers like to do to create customized protocol. And at the same time, it's also hard to adapt the software for custom needs as well. And we also find some of this proprietary software, they can be quite costly, and they also inhibit collaborative development. So as such, we decided that maybe we can go a one level lower than what this package is offering, to create a software package, that can help us to create protocols and pick lists, for some of these commonly used liquid handling. So we started to adapt the software package developed by the Edinburgh Foundry called Plateo, that will help us to enable the creation of the protocols and pick lists. Okay. So the last package, moving forward, what we want to do is really to integrate the current lab automation software with the other libraries that's already available, to enhance our data tracking capability. So last but not least, we have the codon usage table, so sort of to provide a standardized means, okay, for the usage of codon usage. Okay, so this is by the Manchester and the Edinburgh group. So essentially, we view this as a starting point for the software development projects within the BioFoundry, right? So we are not saying that these are the only 3 modules that will be developed, but we hope that we form a sort of a framework, for people to contribute. Eventually, we like this whole Synbio-Python to be like BioPython, right? Whether it will be the go-to software site for everyone. So with that, I will end with a summary. So essentially, we have developed the Synbio-Python as an initial effort, right, to create a standardized open source Python library. So whoever is interested, please contact us through this email. In fact, we have even created an email called synbiopython.org. right? So we hope to have more people joining us to develop novel approaches. So with that, I acknowledge the various funding agency that funds the different collaborators and contributors. And these are some of the useful links you can get hold of the software, right? So with that, I thank everyone and welcome any question.
Thank you, Chueh Loo. That's wonderful. Yep, we can have 1 minute of questions. James has asked for any examples of protocols or experiments that have been implemented with the automation library, and curious about the range of experiments that it can be used for. Like, is it limited to certain machines, liquid handling robots?
So I think right now what I understand from Peter is it can be used for echo, the lab side echo. And I think he's also developing it for the OpenThrone as well.
Cool.
Right. But what I understand from him is that it is easily extendable because it's highly programmable, right?
Costas would like to know, do these tools require input, collaboration from the companies constructing the instruments?
So right now, I think we have to sort of have an understanding of some of the APIs that's been used by the instrument company. For example, when we are trying to develop it for OpenThrone, we actually contact OpenThrone, like in terms of how can we access certain sort of functions and API, right? And then, but the end result is that we decided not to touch their API, right? But I think that there needs to be certain communication between the companies.
Okay. All right. Thank you, Chueh Loo.
Thank you.
So we have time for at the moment. If there's any more questions for Chueh Loo, he can just pop them in the Q&A box and he can answer those at his leisure. Can you?
Thank you. So I'll stop sharing now?
Yes, please. Somehow. Thank you. And Esteban, you're welcome to bring up your screen now.
So, hi everyone. Can you confirm that you can hear me and you can see my screen?
Yes, that's fine.
Awesome.
Yes.
Thank you. So, well, first of all, I would like to thank Matthew and Nat for the kind invitation to present our work, which really is about trying to boost learning in biofoundry through testing, and how do we integrate systems biology and various omics into the pipeline. And this is not working. Okay, so many of you have heard of the Amyris project, where they're basically trying to make 450 molecules, and the goal of this is to actually get these molecules from milligrams to kilograms. This, you can find the details on the publication from Amyris. And what we've been asked is really to complement Amyris Biofoundry with systems biology. And we've been given two difficult chemical targets. And our proposition is that by increasing the test that we do in those strains, we can reduce the number of iterations to get to the target. So we were given 500 constructions, so basically 250 strains per chemical. So the key question, or what we are trying to answer, is in the context of biofoundries, is there a role for systems biology? So we've heard many times, and particularly in industrial genome foundries, they tend to suffer from poor learning, and a lot of the scientific information is lost. SNP information is very sparse, and phenotypic changes may only be observable in the presence of other modifications, and we miss many of those. So the other thing is we suffer from a lot of prior data, and if we actually look at what data is available, it's very, very scarce. So our value proposition is that by integrating a full systems biology data set into the capabilities of the Amyris biofoundry, we can complement the learning component. And why is this useful? So this is what Amyris typically do. They will grow cells in a 96 well plate. They will do endpoint analytics and then classify the strains as the high versus the low producers. Then they will do typically next generation sequencing, get the SNPs, and that enters the next round of design. And as we've heard and seen in the farnesene project, that took a large number of iterations to get to the target goal. So our proposition is that by using bioreactors instead of plates, by increasing the analytics, so measuring metabolites intracellularly, measuring proteins, and increasing the getting data for flux analysis, we can actually increase the knowledge and get to less number of iterations to get to the desired phenotype. So as we can see here, we're hoping that our approach will result in less number of iterations. Of course, with this, we encounter a large number of challenges that we had to solve. For example, reproducible characterization of large number of strains, which of course we're not very used to. And we need to get omics extractions, which is normally not something we do. So we need to have access to larger volumes. So a 96 well plate is not going to do the job here. And then, of course, when you extract this very large number of samples, you need to prepare and process this large number of samples. And the big challenge, of course, is we generate enormous amount of data, and how do we handle this data? And once we get all this data, how do we store this data in a meaningful way that we can then use this data to learn and design the next set of strains? So the first challenge is typically we use plates, and they're great because they allow us to do very high throughput. They're very cheap, and they allow us to do short cultivation. They, of course, have limited control, and they provide very little data apart from growth. We don't really get much more. And this was obviously not an option for us, because even deep well plates were not gonna enable us to do the omics that we wanted. The difference with bioreactors is, of course, they're highly reproducible. We have very controlled environment. We can have different feeding strategies, so mimic real life design, a real bioprocess. And this is very important, because what we typically do is, once we select a subset from the plate, we then move into bioreactors. Of course, bioreactors have a disadvantage. They, of course, represent a high capital cost and operational cost, and we end up with much longer cultures, because we can do much longer fermentations, with a lot more substrate. And of course, the throughput is limited by the number of reactors that we'll have. In our proposition, strict control of operation condition was needed, and without a proper fed batch, our value proposition was not possible. And we really had a big requirement of high quality reproducible data, so we didn't have to do biological replicates for each culture. So the first, obviously, thought was, okay, would an Amber system work? And that was obviously the original system of choice. But of course, as many of you that may know, it comes with a very, very high operational cost. And it wasn't really fulfilling our needs for automated sampling. And indeed, we couldn't even take any samples for omics. So it's great for media development and all this, but I guess it's more catered to large biopharmaceutical companies. So we went with an Infos biosystem, bioreactor systems, where we have 16 bioreactors, which They're small, they're up to 0.5 liters, which is the perfect volume that we needed. They have online OD monitoring. We can obviously control temperature, pH, dissolved oxygen, everything. And importantly for us, we could integrate the system to GCMS for online off-gas analysis. And they came with a very nice software that facilitates integration into our LIM system. With this system, we can run up to 16 designs a week, and we were given 2 years to complete this project. So this will fit nicely with our needs. So what type of data do we get from using bioreactors that we don't get from plates? So for example, all this data gets fed. We get a plethora of data, including, for example, certain products. Our bioreactors are, as you can see in the picture here, connected to a mass spec. So in the mass spec, we can sample certain products. So in this particular case, for example, we use RQ to control our bioprocess, which of course cannot be done without this mass spec. And additionally, we get lots of soft sensors. For example, we can calculate biomass online using some of these multiple soft sensors that we can introduce. So basically, AXA, who's pictured here, did a lot of work optimizing the process development. So we're feeding up to 500 liters of sucrose to our bioreactors. We start with a fat batch, then we move on to a fed batch, which obviously starts automatically as soon as something is triggered. And we take 4 samples across 48 hours, and then we let all the sugar to be consumed. And then we take supernatant for extracellular metabolites. We take a cell pellet for intracellular metabolomics and proteomics. And we end up with about 10 to 20 samples per strain, which then needs to be processed. So as if you do the maths, these end up with lots and lots of samples that need to actually be processed at the same time. So the first thing we did is try to automate sample preparation. So for the metabolomics, we felt that full automation wasn't very feasible because we have lots of variable protocols. We have a lack of standardized workflows, and we needed to find a robot that would allow us to do that. So we went with this basically robotic pipetting Integra systems, which facilitate simple workflows. So for example, we can do organic extraction of metabolites from Eppendorfs to plates or HPLC vials. We can use this for Bradford assays to quantitate the extraction of proteins. We can do dilutions of standards, etc. And it's reasonably easy to manipulate from 96 channels into vials. We also have a Flowbot robot that allows us to move liquids from plates into vials, etc. So we can actually move our samples into our various analytical platforms. So once we have developed a reliable protocol for intracellular metabolomics, which of course is a big task, we went with basically what is in the literature, which is intracellular metabolomics extraction using quenching, and we use the abundance of label metabolites such as NADPH, NADH, and ATP as a proxy to evaluate whether our extraction protocols were working. And then, of course, the next step was to get reliable analytics. And for that, we spiked all our samples with a fully labeled internal standard, which is basically the same culture grown on universally labeled glucose. and this allows us quantification without the need of injection of a standard curve. This was very important not to have to inject all the samples at the same time as we were used to do. As many of you would know, LCMS suffers from ion suppression, and depending on the matrix that you have, you will get different counts depending on the day and the state of how clean is your instrument on the day. So by spiking your samples with universally labeled internal standard, we can get rid of this bias. And then Manuel and Tara spend many hours optimizing and developing methods. Out of the 62 metabolites that are involved in in one of the molecules, for example, we can quantitate 58. And we spend a lot of time developing these methods to get to this point. The next stage, of course, was to develop a protocol for high throughput sample preparation for proteomics. So we went with a Starlet robot, and we're using the STRAP workflow. We base this on the use of positive pressure, which is cost effective. We can run up to 296 well placed per day. So in a run, we use very little tips, so only 2 tips are used per sample, and we can process about 1 plate in 4 hours. So within a day, we can get more than close to 200 samples per day. The method returns a sample plate for drying down, and in this case we looked at automation, but that would be way too complex. This will require heating, and that will degrade our samples. So basically what we do is we take the plate out of the robot and manually load onto a vacuum centrifuge. With this pipeline, we can measure up to 2,500 proteins in a 60-minute run in an Orbitrap. And of course, it was important that this includes all the introduced proteins in our pathway. The next bottleneck, of course, was machine time, and this was the work of Gert and Liane. And what we did there is basically all our LCs contain 2 pumps where we can do switch of columns. So our run is 60 minutes, and then what we typically do in many analytical facilities is we spend a long time cleaning columns. So by having these dual pumps, we can really every 60 minutes get an injection, as opposed to close to 90 minutes that we were doing if we did not had this dual column switch system. And then, of course, the next challenge was, of course, how do we handle and store the data that we generate? So logging of a large number of samples, and then we had a bit of a mess at the beginning with samples going missing. And then we have to communicate this with many people, keeping track of the progress, and mapping the results back to sample IDs was very difficult. We had to collect data from fermentation, metabolomics. We had data coming from GCMS, UPLC, LCMS. We had very large files coming from our Orbitraps. And then, of course, all of this data needs to be fed into a genome scale model so we can actually analyze the data. So that was a big challenge. So the first thing we did is, how do we do this more efficiently? How do we deal with from sample submission to obtaining the results in the required time so we can get everything done? So this was the work of Robin and team, where they basically got a very nice data warehouse plus data submission system that allows us to keep track of all the data and store the data safely. So what we did for data handling solution is we're using Smartsheets, which allows us to get all metadata information for processing. So for example, the client will submit a form online, and then we can see everything on our end. So we can see this analysis was assigned to who, which instrument, when it came, when the analysis was completed, if the client or the person submitting has certain notes, and what do we do after the analysis in terms of data processing. Then, of course, the big challenge is how do we store all this data that is coming? And for this, for data warehouse, we're using an open software called OpenBiz, which samples are assigned a unique ID number, and it allows us to run a list for analytical automation, and this minimizes operations errors. So it pulls all the data from multiple sources. So for example, the data coming from our bioreactors, which is a third-party software called Eve, and all the data coming from all our omics results and all our bioanalytical instruments had to come to one place. So the data is stored as files, and you can actually get information about the fermentation, the media preparation, the reactors, and analytical data, which of course, without this, it would have been impossible to get this into one unified Excel spreadsheet for modeling. So really what we learn is that OpenBiz facilitates data retrieval for data and metadata associated with all the samples. And this, of course, is a massive task, particularly if in the future we are to use algorithms, for example, modeling or machine learning. Just to get one table that contains all this information is a massive task if this information is captured in multiple platforms and coming from multiple systems. So with all this in place, of course, the next challenge was how do we actually design the next generation of strain, and how do we make sure that we learn from all this data? So we, in our group, we are very good at metabolic modeling. So we use traditional flux balance analysis, which basically is a collection of matrices that are solved, which represent the data from the genome, where we have our reactions and our metabolites, and we run a simple mass balance assumption, which is steady state, for which was important to get all our fermentation data reproducible. Then we have constraints which can be imposed to the system, for example, thermodynamic feasibility of our reaction or the capacity of our cells. So this basically is the omics data that we use as constraints for our model. Most of the design work was done by Tim McCobbin, which is basically the soul of this, together with AXA, the soul of this project. So when we design things, we basically, we have models which are, where we have many degrees of freedom. For example, in this very simple system where we're going from substrate to product one, we have on the overdetermined system where we have many degrees of freedom. So in FBA, we don't have a single solution. And we have a feasibility space, which is defined by the constraints. So the first constraint is, of course, the mass balance constraint, but this, of course, becomes a very large space. Then we have our thermodynamic constraints. So for example, for our reaction to be feasible, we need to have a negative delta G. So we can get this convex cone, but still infinite. And then, of course, we have the capacity, which is basically further constraining our system so we can actually find a better solution. So stoichiometric models are good at predicting overall metabolic trends, but they lack mechanistic information. And many times we lack this mechanistic information, which is needed to better define and get a sense of the sample elasticity, which is the degree of saturation of our pathway. So metabolic models can be expanded by considering enzyme kinetics. And for that, we require metabolic measurements, so intracellular metabolomics, and we require protein abundance measurements, which we were using from proteomics, and requires restriction of scope of model to relevant pathway to be engineered. So what I'm trying to say is typically a flux is a function of two things, the driving force and the capacity. The first one, the driving force, can be constrained by thermodynamics. So do we know the thermodynamics, is this reaction feasible or not? And the capacity is a function of the enzymatic abundance. And most of the time, increased flux does not necessarily means increasing pathway expression. So to do that, we need to fine-tune protein expression level and metabolite concentration. So how do we do that? We have a lot of regulation in cells. We have basically metabolism is a coordinated effort of thousands of reactions happening at the same time inside a cell. So how do we balance thermodynamic driving force with these constraints? So, and the other thing is we need to maintain certain toxic metabolites low to actually drive thermodynamics. So to do that, we use kinetic models, and for that we require enzyme kinetics, which are typically unknown. So what we did is we measured values which typically or arguably do not reflect in vivo concentration. And for that, we require data estimation. We need to make sure the feasible kinetic parameters and samples to give a population of potential models. And this ensemble model is used to make predictions or refine by screening for relevance against other conditions. So what I'm trying to say is the kinetic properties of an enzyme will define the sample elasticity or the degrees of saturation of a particular pathway. So our goal is simple. We want to optimize flux. We know flux is a function of driving force time capacity, and our value proposition is driven about data-driven science will enhance and reduce the number of iterations. So we can measure flux, we can measure thermodynamic constraints by metabolomics, and we can measure protein abundance. So we can construct these kinetic models, will enhance our learning. And with this, we can design overexpression targets for our next round of design. So what we normally do is when we're designing a single strain, we need to measure flux, metabolites, and we sample thousands to 10,000s of models that can suggest behavior. And then we do metabolic control analysis or MCA to predict rate limiting step. So from there, we will build the next generation of strain to test the hypothesis. Again, we measure flux, metabolites, and enzyme capacity. We prune model ensemble using rejection sample, and we use MCA to formulate new target. And we do this over and over until we reach our kilograms. So does this work? So let me just illustrate some of the things that happen in a case study. So if we call our first product production of X, so this is what a simple real pathways looks. And as you can see, this is highly complex. Normally we have biomass strains. We have complex reaction mechanisms. In our case, we had a P450 and a PKS for one of the enzymes or steps. We have enzyme promiscuity, and we have certain toxic intermediates that obviously kill our metabolite. We have metabolons, we have allosteric regulation, all of these happening. And then we generate a ton of data that is all stored in OpenBIS with our modeling data, with our proteomics data. We have a reference strain, and then the best we can do sometimes is just these heat maps and try to infer changes, and then we know we can map all this to a pathway, and we have to deal with massive data sets. But eventually what we're trying to do is go to the next design of the next plate, which will be the next set of 96 strains, which would be the next one. So does this work? So what I'm showing here is a very small screenshot of in this particular model, we had 9 reactions. So we can see that the kinetic model identified 2 key limitations: the lack of availability of acetyl-CoA. This was missed by Amyris because it's outside of the original design scope. And then the second thing that our modeling showed is a P450 limitation. This was shown by flux control coefficient, so P450 and acetyl-CoA. So are the reactions that are having the highest control over product flux, which is reaction 7. So what we're trying to do is maximize for reaction 7, and this basically points to the two problematic reactions. Concentration control coefficient also indicated acetyl-CoA has the highest control over reaction 4. So with that, we went back and moved to the design of our next set of strains. And what I'm showing here is 96 design, so basically a plate where we actually took this information. We took the acetyl-CoA limitation, we tried to remove this, and we built and observed that we could reach up to a 9-fold increase relative to the next improvement. And so if we use this approach, we can actually show that learning in biofoundries can be enhanced through systems biology. So we have established protocol for automated high throughput characterization of strains in bioreactors. We showed that systems biology's approach can enhance the learning cycle of biofoundries. However, all this comes at a very high cost because omics are very expensive. And it needs to basically be balanced whether the cost of generating more strain versus generating this type of data needs to be carefully evaluated. So with that, I would like to thank all the people that did this work. So obviously the building part was done at Amyris. The design, as I mentioned earlier, was done primarily by Tim under the guidance of Lars Nielsen. All the testing was done by our amazing Bioplatforms Australia team at AIBN, and most of the work there was carried on by AXA, and I hope, and the learning was done by all of us, and I hope you've learned something from listening to this talk, and thank you.
Thank you, Esteban. That's great. We've got a few questions, so I might let the speakers ask the question in person. So Maciej, I'm going to turn on your microphone if that's okay. Maciej Hołowko.
Hi, can you hear me?
Yes.
Perfect. Hi, Esteban. I was just wondering, how would you compare this approach where you use full-scale bioreactors to where you would use microbioreactors? So I'm talking about like a biolector system.
Yeah. So as long as you have enough samples to do the omics, which wouldn't be possible in a biolector system, this approach wouldn't work. You do get all the data in terms of the control of the fermentation, that for sure, but the main problem is the amount of samples. So to do 4 or 5 sampling from your bioreactor, you do need larger quantity of samples.
Mm-hmm.
So that would be the limitation.
Okay, I see. Thank you.
Thanks, Maciej. Nathan, I'll go to you now. Just give me a second to turn your microphone on. Nathan Hillsong.
Yeah, this is fantastic. Thanks so much, Esteban and Chueh Loo, for your presentations. Maybe just a couple of quick comments, Esteban. I think lots of opportunities for collaboration around metabolomics and proteomics sample prep automation. Chris Petzold, one of my colleagues, might have another question for you. Also around maybe comparing results from the Infor's bioreactor system with Amber's. But my question is really around if Amyris gave you 250 strains per each of these targets, how did you decide between exploration and exploitation, and how many cycles of engineering did they give you?
Yeah. So I guess that was one of the things we had to adjust. So each of our design was a plate. So we had to submit 96 design per round. So for us, it would have been better to be able to submit less design. So some of we were constrained by 96. So sometimes we had to fill the plate with some design of experiment, which is obviously not ideal. If we were able to submit less samples, it would have been a lot better. So to answer your question, we were given basically 5 rounds of design. So roughly 5 rounds of design. Does that answer your question?
Yeah, it does. That's perfect. And maybe Chris Petzold will have a follow-up question.
Yeah, sure. I'll turn on Chris's microphone. Chris.
Hi, thank you. Esteban, could you tell me what would you say the biggest bottleneck or bottlenecks are when executing an experiment at the scale?
Sure. So I guess a big bottleneck is we can only run 16 bioreactors per week. That is one bottleneck. Then out of that, I mean, so even if we run this, AXA will spend maybe 2 months doing, utilizing the bioreactors 24/7. We then generate very large number of samples that then need to be processed for proteomics and metabolomics. And then processing all these samples, I think AXA and the team did an amazing job getting all this fully automated. But eventually it all comes down to, for example, for proteomics, just to give you an example, we still need a 60-minute run per proteomic sample. That only leaves us with 24 samples per day with a fully utilized system. So despite we have 4 Orbitraps, so we're very lucky to be able to run our samples in a relatively short time, then we need to compile all this data into something where if we did not had OpenBees, it would have been impossible to generate that single Excel spreadsheet that team could use and redesign the next set of strains and construct his kinetic model and calculate fluxes. So there are many bottlenecks. To answer your specific question, I would say the biggest one is probably still the bioreactors and getting that of the necessary quality to do the next round of design.
Thank you so much.
Okay. Any more questions? I think we're at time. Oh, there's one more. Your Yaojun. I'll turn your microphone on, and that'll be the last question. Okay, you've got the floor now. Yaojun.
Can you hear me now?
Yes.
So yes, thanks for the nice talk. I just want you to maybe give us some comments that on the directions for secondary metabolites related genome scale modeling, because I know that for the things you are talking about, maybe there are many other public database and data can use. But for some like those natural products related, secondary metabolites related modeling things, like,
yeah. So we've tried, and not for this project, but we've tried to construct these genome scale models for secondary metabolites, and it is actually difficult because the secondary metabolite production is not growth coupled. No. So many of the algorithms that we typically use to design strains rely on this. So when you don't have this growth coupling, it makes it challenging. Having said that, genome scale models are a very useful functional annotation of the behavior and the capacity of your system. So you do get a lot of information from them, but the nature of secondary metabolite production creates certain challenges compared to when you actually work with something linked to that is growth coupled.
Yes, thank you. Yeah.
Okay. Well, thank you, Esteban, and thank you everyone for the questions. We're well and truly out of time now. Esteban, would you mind turning your screen share off?
Absolutely.
I can't find the button. Cheers. Thank you. And thank you, Esteban, very much, and Chueh Loo for your talks today. I think everyone's really appreciated it. And Matthew, happy speedy recovery from your taste of COVID immunisation. Okay, everyone, well, take care. So nice to have you all with us today, and look out for next month, which will be a European time slot, but 2 equally fabulous talks. So thank you for our speakers. Virtual clap, and we'll see you all again soon.
Yeah, bye.
Bye.